All right, let's just hop in here. So this is lab 3. So lab 3 is about cells. So cells are the smallest unit of life. All living organisms are made up of cells. Organisms can be made of cells in a unicellular fashion, which is just one cell, or they're multicellular, have many cells, like millions or billions. And just like your body, all cells contain smaller parts known as organelles. So there's 2 types of cells. There's eukaryotes, which are more complicated with membrane-bound organelles, and there's prokaryotes, which are more simple like cells. So prokaryotic cells are usually unicellular, and like I said, very simple. They lack a nucleus, and instead their DNA floats around the cell and usually coils up. So it's a simple structure, and they're very small, so you need a microscope to see them. And some examples are bacteria and archaea. Eukaryotic cells are multicellular. They're complex. They have organelles. Their DNA is stored in a nucleus, and they're stored as chromosomes. So eukaryotic cells you can see with the naked eye, and examples are animals and plants. In a eukaryotic cell, there's many structures, so we'll go through them. So one is the cytoplasm. This is the liquid that fills the space not occupied by the organelles. Mitochondria converts food energy into ATP. The nucleus contains cells' DNA genetic information. The plasma membrane, this is on the outside, this is the phospholipid layer that surrounds the cell and controls what enters and leaves. DNA, this is where the genetic makeup is stored. Ribosomes, these are symbols where amino acids are made into proteins. The endoplasmic reticulum, this is the synthesis of proteins, lipids, and steroids. There's the rough ER, which contains ribosomes. There's the smooth ER, which does not contain ribosomes. The Golgi apparatus, this modifies, sorts, and packages proteins. Then we have the peroxisome, which breaks down fatty acids. We have the lysosome, which, like the stomach of the cell, digests excess or worn out organelles. Then we have the centrioles, which help with cell division in animal cells. So you can look through this on your own. So there's different human body cells. There's lots of different ones. So different cell types have different functions, and their function will determine which structure and organelles they have. So we have, for example, kidney cells, liver cells, bone cells, brain cells, blood cells, etc. Here's a video for you to watch on your own. Okay, so the movement of materials. So the plasma membrane is semi-permeable. So cells are highly selective in what materials they allow in and out. And so what structure allows materials to move in and out of the cell? This is the plasma membrane. And like I said, the plasma membrane is selectively permeable, and only certain small non-charged molecules can diffuse across the plasma membrane without assistance. So passive process or transport, where it moves down its concentration gradient, and materials move from high concentration to low concentration. And so no energy is required. And the example here is diffusion. Active process transport, this requires energy and is used to move the substance against its concentration gradient. So it's moving material from low to high concentration. And since it's going against its concentration gradient, it requires energy or ATP. And the example here is the sodium potassium pump. So passive transport, it's going down the concentration gradient, and active transport, it's going against the concentration gradient, so it requires energy in the form of ATP. So osmosis is the movement of water across a membrane from an area of high concentration to low concentration through a selectively permeable membrane. And the direction of water is determined by the tonicity of the solution inside and outside the cell. So basically, water will follow the salt. So what is tonicity? Tonicity is the relative concentration of solute and solvent. So we have three different types of situations. We have hypotonic, isotonic, and hypertonic. So in a hypertonic situation, water has more of the solute than the cell does, and this causes the cell to shrink because the water is exiting the cell. In isotonic, water and solute are at the same concentration, and so water is exiting and entering at the same rate, and so you have nice healthy cells. In a hypotonic, water has less of the solute than the cell does, and this causes the cells to swell and can eventually burst because water is entering or rushing into the cell. So just some concepts to real life. Why does your skin wrinkle in the bath? Well, you're sitting in a hypotonic solution, and so water rushes in and causes your skin to swell, causing wrinkles. So why does a snail die when you pour salt on it? Well, adding salt water is a hypertonic solution, and so water rushes out of the snail, causing it to lose water rapidly, and thus the snail dies. What happens when you spray perfume or cologne in a room? Well, the smell diffuses throughout the whole room from high concentration to low concentration. So here's another video for you to watch. And then the activities for the lab. So use your resources. You're going to list the functions of each organelles. You're going to draw a eukaryotic cell. You're going to view eukaryotic cells under a microscope using the cheek cell swab. And you're going to describe what you see, label the structures listed. You have another activity to watch on YouTube. And you're going to explain what hypertonic, hypotonic, and isotonic is. And then the last thing is the effects of external environments on cells. So you'll just fill in the table. Okay. So lab 4 then, just to give you a quick review. So remember, tonicity is there's hypertonic, isotonic, and hypotonic. Hypertonic, the water moves out and the cell shrinks. Isotonic, there's no net movement, it's normal. Hypotonic, water moves in and the cell swells. Okay, there's a video for you to watch. Okay, so this lab is about DNA. So DNA again is deoxyribonucleic acid. So this set of instructions found inside cells that tells them what to become, what to do, and when to do it. So the genetic material of human beings and most other organisms, and DNA is found in the nucleus. In prokaryotes, it's usually coiled up and free floating. Okay, so here's the structure of DNA. The DNA molecule is made of 2 strands that complement each other, and this creates a double helix or a double-stranded molecule that looks much like a long twisted ladder. And again, this is known as a double helix. So the chemical basis of DNA. So DNA is a macromolecule built of 4 different nitrogenous bases. So we have adenine that we usually just refer as A, thymine, T, guanine as G, and cytosine as C. So each nucleotide contains 3 components, a phosphate group, a sugar, and a nitrogenous base. The phosphate and sugar molecules make up the backbone, and the bases project to one side. Bases are joined by hydrogen bonds using complementary base pair rules. So be sure to remember this. So A pairs with T via 2 hydrogen bonds. G pairs with C via 3 hydrogen bonds. And that's shown here. So you can see that adenine and thymine, they have 2 hydrogen bonds when they bond together. And guanine and cytosine have 3 hydrogen bonds. Okay, so just to remember, A binds with T and G binds with C via hydrogen bonds. And so here's an example. So you can go through this on yourself. You just want to, what is the complementary strand here? So A goes with T and G goes with C. Okay, so chromosomes are packaged DNA. And so RNA polymerase unwinds the DNA during transcription to break those hydrogen bonds between the G, C's, and the A's and the T's. So DNA function, so cells use the genetic code stored within DNA to build proteins. So DNA is replicated, so it can be transmitted to the next generation. It stores information. DNA can undergo change or mutations to provide genetic variability and determine the structure and the function of the cell. The genetic code lies in the sequence of the nucleotides that make up each gene along the DNA molecule. So to read the code, you must go through two steps. So first is through RNA. So RNA acts as a messenger, carrying instructions from DNA for controlling the synthesis of proteins. They're single-stranded and contain no complementary strand. The ribose sugar in RNA contains an additional oxygen compared to DNA. And instead of the base thymine, RNA contains the base uracil. That means that adenine will pair up with uracil during the protein synthesis process. So A will pair with U when going from DNA to mRNA. So just remind you, so the differences between DNA and RNA. So the sugar in DNA is deoxyribose. You can see here there's a hydrogen, whereas in RNA it's ribose, and there's an OH group there. And then the base is, in DNA, there's a thymine, and in RNA, instead of thymine, it's uracil. So just some more differences and similarities of RNA and DNA. The similarities are they both are nucleic acids. They're both composed of nucleotides. They both have a sugar phosphate backbone, and they both use 4 different types of bases. So the differences here is that DNA is found in the nucleus, while RNA may be found throughout the whole cell. DNA stores genetic information, where RNA assists in the processing of genetic information. In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. In DNA, the bases are A, T, C, G, and in RNA, the bases are A, U, C, G. DNA is double-stranded, whereas RNA is single-stranded. And then DNA is transcribed to produce RNA, and RNA is involved in gene regulation and is translated to produce proteins. So there's 2 steps for gene expression. The first step is transcription. So DNA is converted into RNA. So there's a synthesis of an mRNA molecule using the DNA, and this is done in the nucleus. And then there's the second step, which is translation, where the mRNA is converted to protein. And converting genetic information into proteins uses a codon table, which I'll show you in a sec. And the location of this is the ribosome and the cytoplasm. So just to reiterate, there's 2 steps in gene expression. One is DNA transcription into mRNA. The second is mRNA translation into protein. Okay, so again, the first step is to transcribe the gene. So to transcribe, the sequence of nucleotides in DNA is copied to a sequence of nucleotides in mRNA that we call the transcript. In the second step, or translation, to translate a different language, mRNA is translated into a sequence of amino acids with the use of the genetic code. So we're going from DNA to RNA to protein or polypeptide. So here's the genetic code that's been cracked. We have the genetic code. So 3 base sequences in the mRNA are called a codon. So 3 bases equals a codon, and each represents a specific amino acid. So there are 64 possible codons, 61 refer to an actual amino acid, while the remaining 3 are stop codons, which signal the protein to terminate. And then one of the codons, AUG, stands for the amino acid methionine, where this is the start of translation or signals the beginning of the polypeptide synthesis. So codons, these cells decode mRNAs by reading their nucleotides in groups of 3 called codons. Here's a couple features of codons. Most codons specify an amino acid. There's 3 stop codons that mark the end of the protein. There's 1 start codon, AUG, that marks the beginning of a protein and encodes the amino acid methionine. And then codons in mRNA are read during translation, beginning with the start codon and continuing until a stop codon is reached. And then this is done in a 5' to 3' fashion. So how to use a genetic code chart. So first look for AUG on the mRNA strand, and then proceed in units of 3 until you reach a stop codon. So an example here is, here's the DNA strand. It gets transcribed into an mRNA. So you look for AUG, and then the next one would be CGU. The next one would be UAA. And then if you look at the genetic code, AUG is methionine, CGU is arginine, and then UAA is stop. So the protein would be translated into met, arginine, and stop. And this is what we call the central dogma, or the big idea of biology, which is DNA is transcribed to RNA, RNA is translated into protein. So DNA stores the information to run the cell, RNA functions to make proteins, and then the proteins actually do the work in the cell. So just to give you a review of gene expression, number 1, DNA in nucleus serves as a template for mRNA. mRNA is processed before leaving the nucleus. mRNA moves into the cytoplasm and becomes associated with ribosomes. tRNAs with anticodons carry amino acids to the mRNA. The anticodon-codon complementary base pairing occurs, and then the polypeptide synthesis takes place one amino acid at a time. So what's important to remember here is that you start in the nucleus doing transcription, and you end in the cytoplasm doing translation. So there's a review video for you to watch. So what you're going to do in the lab report is you're going to decode a sequence using the genetic code. So make sure to read through the introductory material in the lab manual and then complete the activity. Then you're going to draw a picture illustrating transcription and translation. And then you're going to watch this strawberry extracting DNA video, which you can try on your own in your kitchen. And then you have some review questions to answer. Okay, are there any questions before we wrap up today? All right, that's it today. You guys have a good day.
Thank you.